Our third group of organic molecules is called proteins. And proteins have many different structures that we're going to talk about at length. But if I were to ask you what a protein was, you probably would say meat. That's typical response because that's where we get our dietary proteins. However, we actually take in dietary protein, break it down into its monomers, and use those monomers to build the proteins within our bodies, which are so many things within your body. So many things within your body, so much more than just meat. Um, some of the examples of proteins that we're going to talk about support. Collagen is a protein literally holding you together. Your metabolism that we're going to talk about at length in chapter 6 is controlled by molecules that we're going to call enzymes. These are protein molecules that orchestrate the reactions that are occurring within your cells. Very important. The hemoglobin in your red blood cells that's responsible for holding the oxygen molecule as the blood moves through your body. That is actually a protein. Your antibodies that protect you from foreign invaders, part of your immune system, those are proteins. Insulin, a hormone, is a protein. And last but certainly not least are muscle fibers. These are actually proteins. So let's talk about the monomer of a protein. Remember, these are macromolecules made of subunits called monomers. So our monomer is called an amino acid. Amino acids, there are actually 20 different amino acids that make up the proteins in your body, and they all have these 4 parts. So an amino acid has 4 parts. It starts with a carbon right here bonded to a single hydrogen. An amino acid has this structure right here, which is an amine group that has the nitrogen right there. An amino acid has a carboxyl acid group right here. The amine group and the acid group are functional groups right here. So here's our acid group. And then it has this part of the molecule right here that's indicated with the R. For right now, we're just going to call that the rest of the molecule. Some books actually call it a side chain because it kind of hangs off the side here. We'll talk more about that in just a moment. But this is the structure of a single amino acid, which is the monomer or the building blocks for proteins. There are 20 different amino acids. So logically, if we take a look at the amino acid, all amino acids have these 4 parts. So what makes one amino acid different from another amino acid would be what's sitting in the R position right here, what this side chain actually consists of. So the unique properties of each amino acid would depend on what's right there. Okay? So here are our 20 amino acids, and no, you do not have to memorize these. I show you this illustration just to prove that I'm not telling you a story, that here are the 20 amino acids, and if you'll notice in the purple area right here, all of them are exactly the same. The carbon with the hydrogen, the amine group, and the carboxyl group right here. What differs between the amino acids are what is in the R group right here, or the side chain. So let's take a look at these. The amino acids right here at the top of the illustration, they're illustrated in yellow. Those R groups are hydrophobic. The amino acids right here that are illustrated in a greenish color, those are all hydrophilic. And these R groups right here are either basic or acidic right here. So each of these R groups has properties associated with it that are very important. So when we bond 2 amino acids together, let's take a look at that. When we bond 2 amino acids together, it's a covalent bond that forms, and it's a special type of covalent bond called a peptide bond right there. Again, this condensation synthesis right here with our water coming out, and we now have this monomer, this amino acid, bonding to this polymer, which right now only consists of 2 amino acids bonded together. but where the bond exists right here, that's where the water actually came out of the reaction. So what do we have right here? Take a look at this. There's one amino acid, there's two, there's three. And if you'll see why the R groups are called side chains, because the amino acids actually bond together right here, and the R group just kind of hangs off the side right there. Peptide, this is called a peptide bond. I want you to remember, or write in the margin of your notes, that the word peptide refers to proteins. Anytime you hear the word peptide, I want you to think protein. As a matter of fact, when we bond several amino acids together, very much like when we bonded many monosaccharides together, we called it a polysaccharide. When we bond amino acids together, we're going to call it a polypeptide. So this is a chain, a polymer of amino acids joined together by a peptide bond. So this is starting, we've started a polypeptide here, okay? And a protein then would be one or more polypeptides. Okay, so let's take a look at some various shapes of some proteins. And you don't have to remember these. I just want you to see that they come in lots of different shapes. But I'm not going to hold you accountable for knowing the shapes of these proteins. This is collagen. It's a rope-like structure. This is what your cartilage and your tendons are made out of. Very, very strong structure. These are antibodies. They're kind of a T shape here. These are enzymes. Notice that their structure is very different. And this is DNA polymerase that's responsible for putting or building your DNA, putting the nucleic acids together. Don't forget our discussion of shape or structure determining function. Very important that protein shape be exact. If the protein shape is not right, the protein cannot do its job. Protein molecules actually work by fitting into other molecules, very much like a jigsaw puzzle, so that if they don't fit just right, then the reaction or whatever's supposed to happen doesn't happen. So protein shape is extremely important. Let's talk about where that shape comes from. How does a protein end up to be this shape or this shape? How is that determined? How is this polymer of amino acids, a linear arrangement, end up looking like something like that? Let's start by, let me ask you, how many letters are in the alphabet? And most of you know that there are 26 letters in the alphabet. When you stop to think about how many words you can make out of those 26 letters, it's almost infinite, realizing that there are many different words of various lengths. But we have to think about what makes a word a word. If you write down the word eat, for example, E-A-T, how do we know that that is the word eat? Well, we know it's eat because there's an E in the first position, an A in the second position, and a T in the third position. A word is defined by the letters in the word and the sequence of those letters. If you rearrange the letters, if you take those same letters and make the word A-T-E, it's the same letters, but they're not in the same order, so it's not the same word. If you take a letter away, if you take the E off of eat and you have A-T, you have a word, but it's not the same word. If you add a letter, B-E-A-T, it's not the same word. So I want you to start thinking about proteins very much like that. There are 20 different amino acids, and what defines a specific protein would be the sequence of amino acids in that protein. You have to have the exact amino acids in the exact order to make that protein, very much like the letters in a word. So this sequence of amino acids that make up the protein is so important that we're actually going to call it the primary structure. The unique sequence of amino acids of a protein is called the primary structure of the protein. So if you take a look at this illustration here, you see that these are amino acids right here, and our R groups would hang off the side there. So right after a protein is put together, and we'll talk about how that occurs in chapter 12, but right after a protein is put together, you have this linear arrangement of amino acids. And keep in mind, there are varying lengths of amino acids, ranging from hundreds of proteins to more than that, many more than that. So what happens to these amino acids once they're put together? Well, these R groups start interrelating and reacting to each other and the environment. Again, remember, we're 70 to 90% water. Some of these R groups are hydrophobic, so they're going to try to get away from the water. Some of them like the water. Some of these R groups even like each other. So if they're close together in the chain, they're going to try to fold and come get closer to each other, maybe even bond to each other. So we have all of these interactions occurring. We have a name for that. We refer to all of these interactions and the structures that exist from that or that result from that. We refer to that as the secondary structure of the protein. This is the folding. Sometimes we have the R groups folding on themselves, or we have some helices here forming, but we refer to these reactions as the secondary structure. That's the folding or coiling of the peptide, which ultimately will end up in a final three-dimensional shape, and we refer to that as the tertiary structure of the protein. So keep in mind that this structure is a result from the interactions of the amino acids and the R groups right here, and those interactions are a result of the sequence. So the sequence determines the relationships which determine the final three-dimensional structure. If you change an amino acid up here, it's going to change the relationships between each of them, which is going to change this, which will change the tertiary structure. So you're not going to have the same protein. You're not going to have the same shape. It's not going to do the job that it needs to do. Some proteins are made of more than one polypeptide. So what you would have would be more than one of these folding, which is what we call the process of this shape building. We have more than one polypeptide folding to form a protein that has two or more polypeptides. We refer to those proteins as having a quaternary structure. That's when the overall protein structure that results is a combination of two or more polypeptide subunits. These proteins are very, very sensitive to their environment. Again, this folding process occurs in a very specific environment. There's a very specific pH, a very specific temperature that these proteins will fold correctly. If that temperature or pH changes, then you change the environment. You can change the relationships that form. So shape depends on physical or chemical environment that the protein is folding in. If you change those things, it can affect the structure, and we call that denaturation. That's when you have a pH that is not right or a temperature that is not right that causes the protein to change its relationships or bonding patterns, and it loses its shape. It loses its ability to work. It can't do its job any longer.